It's wonderful to be in Madrid, and I'm excited to share our work with this wonderful audience. So my lab is broadly interested in this, this sort of what we call the nexus of membrane biophysics, and that is the detailed and complex, comprehensive lipidome that is present in all cells, but in particular in mammalian cells, and the consequences of that lipidome on the physical properties of the assembled bilayer. And ultimately, the ultimate interest is, of course, how the composition and physical properties impact on the functional phenotypes, and that is the ultimate function of proteins in the membrane. So today, hopefully I'll have time to tell you two stories. One that Daniel already mentioned, which is essentially how the mechanisms and the determinants by which proteins partition to ordered membrane domains, and a second one that's new and hasn't been published on the asymmetry of the plasma membrane in mammalian cells.
Change and lower cholesterol concentration result in increased fluidity.
Quiet. So everybody knows probably already here about lipid rafts. Let's see if so generally speaking, rafts are essentially organization in the membrane, right? Functional membrane domains that are in the context of a mammalian membrane produced by preferential interactions between certain lipids. And the physical-chemical principle behind what we think of as one type of mammalian rafts are the preferential interactions between sterols, saturated lipids, glycolipids, and the net result of those preferential interactions relative to more polyunsaturated lipids is the formation of this sort of still unique and mysterious entity called the lipid-liquid ordered phase, which can coexist with a kind of more typical liquid disordered phase through a wide space of compositions and physical properties. And this kind of behavior can be observed in synthetic membranes and computational membranes, and indeed even in living membranes. So these movies are showing isolated plasma membranes from mammalian cells, and what you're seeing there is the fluorophore that's been doped into those membranes, and what I hope is obvious, well, at least from the movie on the left. Sorry, there's some conversion issues from Keynote. I shouldn't have switched. But at least the movie on the left hopefully is obvious in showing the formation of these large regions of the membrane that are excluding the dye. And these are these liquid ordered domains, which are presumably representative of rafts in living cells. So this cartoon was published in a sort of high-profile review a few years back, and I like to show it not to advance any orthodoxy or dogma in this field, but to give me an opportunity to explain what I mean by the word lipid raft. And so to me, I think of rafts not as things, right? Not as objects of defined size, shape, composition, but rather as a mechanism for organization in membranes, right? So it's undeniable that certain lipids preferentially interact with one another as compared to other lipids, right? So in the mammalian context, that would be those ones I mentioned earlier, cholesterol, saturated lipids, glycolipids, all of those have preferential interactions as compared to more unsaturated lipids. And the net result of those interactions in the collectivity of the membrane, inherent collectivity of the membrane, is some kind of organization. But the specifics of that organization are going to be highly context dependent, right? So, for example, in a membrane that doesn't have very many of those components, boy, that's not going to work, like those ones at the top, these interactions may yield really kind of transient molecular level complexes that probably fall apart on the time scale of milliseconds. But then if there's simply more of those lipids, or if those interactions are stabilized by more specific protein-protein, protein-lipid interactions, then they can be driven to coalesce into larger, longer-lived, and presumably functionalizable domains. Although here, this cartoon, I think, points out something really important, which is that you're seeing maybe a couple of dozen lipids, a few proteins, but certainly that level of organization could be functionalizable, right? That's ultimately how one could regulate the interactions between any two proteins, which is sort of the fundamental unit of signaling in a cell. But those things are still tiny in molecular scale. And then the final kind of end state of these organizations is like really the condensation of what we call the RAFT phase, which we can actually visualize in an isolated membrane, which is what those movies were showing earlier. And an important kind of assumption built into this model is that what we can learn from this terminally phase-separated state about the properties of this RAFT phase are reflective, at least to some extent, about these more functional and physiologically relevant organizational states. So the purpose of RAFTs is easy to imagine. Probably the most widely studied one is in signaling, where, as I mentioned, they essentially can act as little reaction vessels, compartments that will selectively concentrate certain proteins in order to tune signaling reactions at the plasma membrane, for example, in the immune synapse. Actually, the original proposed function for RAFTs was in trafficking, where they would act like a bundling mechanism to deliver a package of lipids and proteins that belong at the plasma membrane all together, right? So like the cellular precursor of Amazon Prime, where they just dump all your stuff into a box and send it to you. But these cartoons, I think, are nice, and they show a useful proposal for how this could work. But I think that we still really don't understand on a mechanistic level, certainly in mammalian cells, ultimately how membrane organization impacts on cellular physiology. And that's because really of a lack of good tools, right? So I showed those synthetic membranes earlier, and those have been an excellent tool to define the physical-chemical principles behind RAFTs, but they are not really good models of biological membranes. They're very simple. They don't have proteins usually. In contrast, living cells are very difficult to study because any perturbation that one does has kind of multifactorial effects that radiate out and are difficult to interpret directly. So for us, a really major advance has been the development of this model system that we call giant plasma membrane vesicles. It's a terrible acronym, but you're going to hear me say it a couple of times, GPMV. So what you saw in this movie, I'll just play it again real quick, are some kind of generic mammalian cells. Basically, all mammalian cells do this. We torture them by throwing some terrible chemicals on them, and they respond in this very odd way by essentially inflating large regions of their plasma membrane as like a balloon, right? So if you've ever seen an experiment with a GUV, a giant unilamellar vesicle, that's basically what these are, except they're made of the plasma membrane. They're sort of the size and shape of a GUV, maybe 10 microns or so in diameter, easily visualizable in a microscope, but they are made, indeed they are formed from the living cell plasma membrane, and therefore they certainly represent the true diversity and complexity of composition of the native plasma membrane, if not the exact composition. But so these are very useful because they're really a nice biochemical prep of the plasma membrane, in contrast to probably all of you prokaryote people. In eukaryotes, plasma membranes are very difficult to analyze because they're very difficult to separate from all the other internal membranes. So this is a very nice prep to do that. Yes?
Is the asymmetry intact?
That's a really good question. I'll get back to it. Really good question. For the moment, I'll say the way you're thinking about it, no. So PS is exposed for sure. But for the RAFT field, one of the most important aspects has been that these ultimately isolated plasma membranes phase separate into coexisting ordered and disordered domains that sort native and exogenous components of the plasma membrane in agreement with the RAFT hypothesis. So this has been a real conceptual and methodological breakthrough for our field. So conceptually, it was really important to show that a complex membrane that is full of proteins, which these are, can still phase separate into an LO RAFT-like and an LD non-RAFT-like domain. But perhaps more important, methodologically, these things give us a really flexible and robust tool to investigate, for example, the composition of these ordered domains in a complex system. And what they're really best for, I think, what they're perfect for, is analyzing this question, which is what makes a RAFT protein? Why do certain proteins prefer the more ordered environment versus the less ordered environment? And for me, I thought this was an interesting question from the very beginning, and I didn't really know why. And then later, I made up a really good reason, which is that the entire function of a RAFT, of a membrane domain, the purpose of a domain, is to specifically recruit some proteins and exclude others. And therefore, the functionality of RAFT domains inherently depends on their selectivity. And so it's important to know which proteins partition in and why. So when we started this work, there was essentially nothing known about why certain proteins prefer RAFT domains. What we had was cartoons like this one from the Albert's cell textbook, or actually this is from molecular biology of the cell. And this implies a really cute cartoon model where the cholesterol-driven RAFT is relatively thick because the acyl chains are forced to straighten out. And therefore, proteins with longer transmembrane domains should preferentially partition there. The numbers sort of make sense. These are reasonable variations in transmembrane domain length. However, the problem was this idea was never tested and raises the question why they put it into the textbook. But anyway, the reason it was never tested is because it's pretty hard to do these kind of experiments in synthetic membranes and basically impossible to do them in living cells. But these isolated plasma membranes, these GPMVs, allow us to directly and really pretty easily test protein partitioning between coexisting phases. And so here's how the experiment works. These are images of one of these vesicles derived from a cell that was expressing a plasma membrane protein that was tagged with red fluorescent protein, RFP, right? So here's the protein. This is a marker for the RAFT phase, so there you see the phase separation. And from there, we can directly quantify the partition coefficient of this particular component between these two phases by simply measuring the fluorescence intensity in the RAFT phase versus the non-RAFT phase. And the ratio is then the equilibrium partition coefficient, which can be turned into a free energy of partitioning if you're a physics type. And the first thing we set out to test is exactly that cartoon from Albert's. Does transmembrane domain length matter for partitioning? To do that, we started with a particular protein. It really doesn't matter at all what it is for the purposes of this talk. All you need to know is it's essentially a transmembrane peptide. A single alpha helix. So all mammalian transmembrane protein are alpha helices. About half of them are single pass like this, so bitopic proteins. And we actually got rid of all of the protein except for the transmembrane domain. And then on the intracellular side, we tagged it with RFP. And then we make various mutations to the transmembrane domain to test various hypotheses, like here in length. So this is what the representative data looks like. The wild-type transmembrane domain for this particular protein has 24 amino acids and partitions approximately equally between the RAFT and the non-RAFT phase, which you can see on this kind of plot, and the partition coefficient is about 1. However, when we shorten the transmembrane domain by just removing 6 amino acids, hopefully you can see that this mutant is now enriched in the non-RAFT region of these GPMVs and depleted from the RAFT region. And you can also see that from the partition coefficient. So then you can do controls, mutate those same 6 amino acids to alanines. That has no effect. You can do that for dozens of proteins. And what is hopefully obvious is that you can see a pretty clear correlation between transmembrane domain length and RAFT affinity, with relatively long proteins having relatively high RAFT affinity and vice versa. So that kind of data can be fit to a very simple model that basically posits an energy penalty for a hydrophobic mismatch, right? If the lipids are long but the protein is short, the lipids have to bend or the protein has to stretch. Both of those things are unfavorable, and we kind of know the approximate values for how unfavorable that is. And so what you see in that dotted line is the fit to this very simple so-called mattress model, using parameters for the thickness of the liquid ordered and the liquid disordered phase that come from sort of realistic simulations. So that was nice. Turned out Alberts was right. Actually, it was a bit of a disappointment. I was hoping he was wrong, but that's okay. Because it very quickly became clear that this observation of transmembrane domain length did not explain everything, right? That simple cartoon wasn't enough to explain all the partitioning behavior that we saw. And we'll just start here. To show that, we made this very simple-minded construct, right? Here's the wild-type protein transmembrane domain. When we mutate it to all leucines, it has the same number of amino acids, the same nominal transmembrane domain length, but hopefully you can see it partitions very poorly to the RAFT domain, right? So clearly there's something else going on. Let me just check if I included this slide. Yes. So one important thing is mammalian proteins are often post-translationally modified by a fatty acid, palmitate. So it's a saturated 16-carbon fatty acid, and that's done by a thioacylation on these intracellular cysteines. When we mutate either of those cysteines such that the protein is no longer palmitoylated, we see dramatic reductions in RAFT affinity, suggesting that this post-translational modification is really important for partitioning to ordered domains. But again, that didn't explain everything, because this all-leucine construct does have those palmitylation sites, and it is palmitoylated, which we checked. So then that told us that there was something else. And basically, we kind of broke down the possibilities into two camps. On the one hand, there may be some kind of very special residue in here, or maybe a motif that binds a particular lipid or induces dimerization or something, and thereby increases RAFT affinity. Alternatively, it's some general property of the amino acid side chains in the transmembrane domain, like, for example, length, that's important. And so to test which one of those was more likely to be true, we simply scrambled up the sequence, right? So all the amino acids are the same, all the side chains are still there, but they're in a completely random sequence from before. And that so-called scrambled transmembrane domain partitioned exactly like the wild type, suggesting that it really is some aggregate physical property and not some sequence-specific behavior. And I'm going to make a long story short, tell you that the first 10 ideas I had for what that aggregate property was were wrong. And eventually, this very talented postdoc in my lab, Joseph, noticed that there was this remarkably robust correlation between the surface area of a given transmembrane domain and its RAFT affinity. That is to say that relatively skinny transmembrane domains that are made of side chains like alanine or glycine, right? You know, a single methyl or nothing at all hanging off the helix, partition very well to the RAFT phase, whereas relatively fat transmembrane domains with things like leucines and phenylalanines did not. And when we noticed this correlation, we realized, well, I was pretty dumb for not thinking of it in the first place, because it really makes a lot of sense when you think about it. And the way we explain it is to conceptualize proteins in a membrane as a thing, any kind of inclusion in a liquid, right? Membranes are liquids. When you put something in a liquid, you create an interface. And interfaces come with interfacial energy, which is unfavorable, which tries to be minimized. So that's sort of trivial, but the key point for this model is that any given inclusion or protein can choose which fluid it would rather be in, the disordered fluid of the non-RAFT phase or the ordered fluid of the RAFT phase. And therefore, there's a differential surface tension, a differential interfacial energy that, when scaled by the surface area, gives you a free energy of partition. So it's simply, the bigger it is, the less well it fits into the ordered phase, because you're just disrupting it more. And it's a very simple linear model. It fit nicely to that correlation we already noticed. That's trivial. But to test it more directly, we constructed a bunch of really totally fake transmembrane domains consisting solely of leucine and alanine side chains that were varied in their number to tune the range of surface area, of various accessible surface areas. And here we get very good agreement between the observations and the model. So wrapping this part up, I hope that I've convinced you that at least in our experiments for these bitopic proteins, RAFT affinity is governed by 3 independent parameters, physical parameters of transmembrane domains. That is their length, their relative fatness or surface area, and post-translational palmitylation. And where we're ultimately hoping to get with this project is towards a really comprehensive and predictive model for any given protein that you might give to us with a sequence that we would be able to predict how well it should partition to the RAFT domain. So I'll tell you that we're not there yet, for sure, but at least for the 100 or so proteins that we've tested so far, this kind of 3-part model seems to do a pretty good job of predicting the RAFT affinity, at least getting close to the measurements. So we were pretty happy about that, and then we were even happier when we realized that all of the pieces in this model can be derived directly from protein sequence, right? You don't need a structure to know how long the transmembrane should be. You just need to count the number of hydrophobic amino acids. You don't need a structure to know how fat the TMD is, right? You can just plug in the amino acids and it spits out a number. And so already we can make a prediction for any given protein, or indeed for all of the proteins that there are. So these are our predictions for all bitopic plasma membrane proteins in the human proteome. And you get this kind of wacky looking distribution. Doesn't mean all that much. It tells you that most proteins are would not be predicted to partition well to rafts, and a relatively small handful would. And so to test these predictions, we took our protein that we started with and picked a few that were kind of near it, and therefore should have similar RAFT affinity, and then 2 that should have very low RAFT affinity. And for those 5, we got our predictions pretty much spot on, in that the predicted RAFT proteins did indeed actually enrich in the RAFT phase, whereas the predicted non-RAFT proteins were completely excluded. What's our timing? Is there a clock? How much? 20 minutes. Brilliant. So then we could go and kind of break down this model and kind of take each of the pieces apart one by one. So for example, I'm just going to show you one of these with the surface area. We can, for our predicted RAFT proteins, right, make the surface area bigger by just flipping out alanines to leucines or phenylalanines. And in each case, by increasing the surface area, we dramatically reduce RAFT affinity. But more impressive is that we can take our non-RAFT protein, make its surface area smaller by turning leucines into alanines, and thereby create a RAFT partitioning protein out of a non-RAFT scaffold. Right. So that was the first part. Now for something completely different. The completely different part is a new project that we're working on, trying to define the compositional, biophysical, and functional asymmetry of mammalian membranes in general, and plasma membranes in particular. So coming back to this idea that composition determines physical properties, which determines function, of course, the non-random distribution of lipids, that is asymmetric distribution across the bilayer, is going to influence all of those parameters and therefore influence the function of a given membrane. And as far as I understand, everywhere it's been measured, plasma membranes are asymmetric in composition between the 2 leaflets, including in E. coli, and bacteria, and mycoplasma. Does anybody know? You should probably measure it. As far as I understand, it's completely conserved throughout the tree of life. So what I mean by asymmetry in mammalian membranes is this kind of particular distribution of lipids where relatively charged lipids like phosphatidylserine and inositol are essentially confined to the cytosolic leaflet of the plasma membrane, whereas the external leaflet contains sphingolipids like sphingomyelin and glycolipids and things like PC. And so that's where this kind of famous cartoon graph comes from. The reason I say graph, I'll get back to it in a second. So the function of this asymmetry is also not very well understood. There's been a couple of implications here and there. A really interesting one is the idea that because charged lipids are confined to the cytosolic leaflet, they really define a uniquely charged surface in the cell. And that can be used for organization, right? So basically, by constructing a cationic protein, the cell can make that go to the plasma membrane or go to a membrane in general. And that's what you see here. These are peptides. These are just GFP with, for example, 8 lysine residues stuck onto the end. And you can see that that goes to the plasma membrane in mammalian cells, whereas just GFP with 2 lysines that isn't as charged distributes all over. So the plasma membrane is highly charged, and therefore that's one way that you could organize it. And then the loss of asymmetry has been very clearly implicated in, for example, apoptosis and death, coagulation. And in each of these cases, well, in a couple of these cases, the exposure of these anionic lipids onto the surface where they don't belong tells other cells there's something wrong with that cell, right? When a cell dies, the interleaflet lipids go to the outside, and other cells know, okay, that cell's messed up, we should eat it. Yeah, mammalian cells do weird stuff like that. But of course, that's really just one implication, right? If we say that each of these parameters ultimately is determined by composition, all of them should be different in a natively asymmetric membrane versus a scrambled up membrane. And basically, we don't know anything about that at the moment. So coming back to this cartoon, it comes from a set of classic studies from Holland, some almost 45 years ago now, where they took red blood cells, which are the only cells that one can reasonably do this in because they only have the plasma membrane, right? They don't have internal organelles, and therefore when you analyze their lipids, you're really only analyzing the plasma membrane. And then they took various phospholipases that would just cut the heads off of the external leaflet lipids, right? And then they would run them on a TLC. They would see how much of a given lipid is left. If they treat it with sphingomyelinase, get rid of all the sphingomyelin from the outside, and then ask what's left. And that's how they would generate these graphs. And the reason that I say graphs is that everyone sees this cartoon, but they don't see the caption, which says this is a cartoon, essentially, right? It's a proposed distribution in mammalian cells. So we thought it might be useful to revisit these very old studies. In particular, oh, that's really ugly. Try to ignore that. Because while these cartoons defined 4 different lipid species, we now know that mammalian membranes are composed of hundreds of distinct lipid species that we can analyze in a really quantitative way by a technology known as shotgun lipidomics, which is essentially just mass spec. So the question is, not for those 4 lipids, but for these 700, how are they distributed in between the leaflets of the bilayer? So that was the old cartoon, and these were our measurements. And I was very annoyed to find that those crazy Dutch people 45 years ago got it almost exactly right by TLC. That's amazing. But maybe I shouldn't have been annoyed because then I would have to explain why we were right and they were wrong. It turns out that they're right, so I don't have to explain that, but it still was worth doing because we have much more detailed information now. And that's sort of what's shown here for the number of unsaturations in all these different lipids, sphingomyelin, PC, etc. And what jumped off the page, and it wasn't that surprising to be perfectly honest, but it really was obvious, this dramatic asymmetry in lipid saturation between the outer and the inner leaflet, where the outer leaflet was composed the plurality of lipids are fully saturated, and the large majority have no more than one unsaturation. Whereas the inner leaflet, the majority of lipids are polyunsaturated. You can see there. Even more interesting, we could go and look at the asymmetry within a particular lipid class, for example, defined by its head group. So here, for phosphatidylcholine, for the various phosphatidylcholines, they're also asymmetrically distributed, but in a really odd and backwards way from the rest of the membrane, where the inner leaflet is actually much more saturated. So the inner leaflet PCs are relatively saturated, whereas the outer leaflet PCs are relatively unsaturated. So I can come back to that for how we think it might work. But moving forward, sort of a cartoon of approximate compositions of the inner and outer leaflet. And the implication is that the outer leaflet is composed practically of saturated lipids and cholesterol, whereas the inner leaflet is composed largely of polyunsaturated lipids. And therefore, one would predict that there would be a significant biophysical asymmetry, not just in charge, but also in things like packing and order that I was talking about earlier. And so we wanted to see if that might be true. And to do that, what we need is a probe that has two properties. One, that it's sensitive to something about membrane properties, right? And there are lots of probes like this, Lordan, DPH, TPA, and we use this particular one called Di4. It essentially works like Lordan. It has a big shift in its spectrum in relatively ordered versus relatively disordered membranes. But actually we found that even better than that, it has a huge difference in its time-dependent behavior as a function of the membrane environment. That is its lifetime, right? The fluorescence lifetime is much longer in relatively ordered membranes versus relatively disordered membranes. And what that looks like in a GUV, you can do lifetime imaging, make maps of lifetime in each individual pixel, and you can see that there's this very striking resolution between the lifetime of this probe in a disordered phase versus an ordered phase. Okay, so that's one thing, but that's easy. The hard part is probing one leaflet at a time. And it turns out that this probe is just right for that, because it's charged. And that charge prevents it from flipping easily across the hydrophobic core of the bilayer, which we've tested, but also it makes sense. So then we can take a cell, we can stain from the outside by just dumping this dye on, and then all we're probing is the outer leaflet of the cell. That's the easy part. Slightly harder, but actually not that much harder, is to get it into the intracellular leaflet of the plasma membrane. And we do that by just stabbing a cell with, with a glass needle. So that's surprisingly easy, and the cells don't die. I don't understand how that's possible, but they don't. And so when we inject this dye into the cytoplasm, the probe partitions into the cytoplasmic leaflet of the plasma membrane and all the other membranes, actually. So this is what it looks like, right? This is a lifetime map of some fibroblast or something that was micro-injected with this dye. And what I hope you can see is that the plasma membrane is stained, and all the intracellular organelles are stained, and these are only the cytoplasmic leaflets, presumably. So the first thing we notice is that the plasma membrane has relatively higher lifetime, suggesting it's more tightly packed than the intracellular membranes. That's exactly what we expected, which was nice to see. The cool part is then we can take that very same cell and also stain it from the outside. And what you see in that case is that the lifetime of the plasma membrane only shifts to higher lifetime. Now both leaflets are stained, suggesting that the outer leaflet has relatively higher lifetime and higher order, higher packing. In a new set of cells, we can stain only from the outside and kind of confirm that these are sort of much more orange, much higher lifetime, much higher packing. So that seems to be robust across a couple of different cell lines we've tried. Even the actual lifetime numbers are basically similar between the two. And then we can go and compare the lifetime as a sort of a correlate for packing to the ordered and disordered phase in phase-separated GUVs. And what we see is that the outer leaflet is indeed pretty much like the LO phase, whereas the inner leaflet is kind of in the middle between the LL and the LD. So now going to intracellular membranes, we can start to probe some of those as well, because after we stain the cells from the outside, after some time, the cells will endocytose their plasma membrane, therefore endocytose this dye, and the dye will actually stain some intracellular organelles, in particular these kind of endosomes. And we can see where the endosomes are by just bathing the cell in some fluorescent stuff, dextran, and that accumulates in these intracellular organelles, and then we can define the membrane of those organelles by just drawing little circles around that, measuring lifetime within those circles, and we can see that the, now can't say outer and inner anymore, right, because this is all inside, but now there's a cytoplasmic leaflet and a luminal leaflet. And the luminal leaflet is still more tightly packed than the cytoplasmic leaflet of those endosomes. Okay, and finally, last data slide. Harking back to the first part of my talk, I showed that relatively skinny proteins preferentially partition into the more tightly packed RAFT domains, relatively fat ones don't. That was all in semi-symmetric membranes, so that's an important artifact associated with those GPMVs, is that they lose some of this native asymmetry of the plasma membrane. But then the question would be, how would this partitioning behave in a membrane that doesn't look symmetric from the outside and inside, but more like ours, that has tight packing on the outer leaflet and loose packing on the inner leaflet? And so to analyze that, we are still not there yet, but one thing we did do is, just in a bioinformatic way, look at all proteins in general and ask if their outer half is different from their inner half. That is, what fraction of the surface area lives in the outer half of the protein. And what we find is that plasma membrane proteins are asymmetric in the sense that their outer part, outer half, is relatively thin and their inner half is relatively fat. Whereas intracellular organelles, like the biosynthetic organelles in the ER and the Golgi, are relatively symmetric. Okay, so to wrap up, the outer leaflet is mostly saturated, whereas the inner leaflet is mostly polyunsaturated. Again, that's the mammalian membrane. These fluorescent probes can image the asymmetric biophysical properties resulting from that, confirming that the outer leaflet seems to be more tightly packed, and this difference seems to impact on protein design, evolutionary protein design. Okay, so I'll finish by acknowledging the brilliant people that it is my great luck and pleasure to work with. In particular, Joseph, who has done most of the work actually I showed today, and Candace, who's sort of my partner in life and in science, and all these wonderful people and the funding. And thank you for your attention.